All right, our second lecture for this week, Lecture 6, Biological Molecules. And by the way, remember the mid-semester exam will cover all the material that we've covered from weeks 1 to week 5. So in other words, everything that we've covered up to date, including tonight's lectures. Okay? So lectures 1 to 6 will be examined in that online mid-semester exam. And one more thing too, while I think of it, a couple of students have asked about a due date for the first prac report, which they've seen the due date. It's in about 3 weeks' time, and they've queried that because they're not doing the prac workshop until 1 week, I think, after that due date. That due date only applies to weekly prac students. Okay, so the weekly prac students, they submit their first report in week 7, I think it is. But for you guys that are doing the weekend workshop, both of your prac reports are due in the last week of semester.
Yep. All right.
Okay.
Yep.
So just in case anyone else is confused about that. All right, so let's go on with Lecture 6, Biological Molecules. And in this lecture, I hope to show you how important a lot of this chemistry that we've been learning is in terms of these major, what we call biological macromolecules that apply equally in whether you're studying plants or whether you're studying animals. The major biological macromolecules. So what are they? The first group are the carbohydrates. Who can tell me what another group is? There's 4 major groups. Carbohydrates is one.
Proteins.
Proteins is another one.
The fats.
Yes, lipids. Carbohydrates, proteins, lipids, and the 4th group are the nucleic acids. The nucleic acids includes 2 major types, DNA, which everyone would have heard of, and RNA, which you may not be familiar with. All right, so let's begin. Organic molecules. Organic molecules are composed principally of the atoms of 6 elements: hydrogen, carbon, oxygen, nitrogen, phosphorus, and sulfur, with the number 1 element, carbon. Why? Because carbon atoms, as we've been learning, form 4 covalent bonds with other atoms. So carbon is incredible in the way that it can actually bond to so many different elements and form a variety of different shapes, chains and branches and ring structures. So carbon, of course, is the number 1 element when we're talking about organic molecules. The covalent bonds that we find, in particular between carbon and hydrogen, are energy-rich bonds. And we'll talk about extraction of that energy in living organisms in more detail in living systems too. There are 15,000 different unit or organic molecules known in living organisms, but only 29 are needed to describe the basic biological molecules. And we'll talk more about these different unit organic molecules also in living systems too. All right, let's just consider briefly the synthesis of these large molecules, biological molecules, macromolecules. They're large molecules composed of repeating units, and those repeating units are assembled by what are called condensation reactions. One condensation reaction in particular is called a dehydration reaction. So you can imagine that a consequence of a dehydration reaction is the loss of molecules of water. That makes sense? Dehydration, loss of water. During a dehydration reaction, small unit molecules, which we call monomers, and think of these as building blocks, if you like. These monomers react together to form larger molecules, which we call polymers. So we can think of, just very simply, we can think about the building blocks as just individual monomers or subunits. And then if we actually join them together, if they were bonded together, then we would have a polymer. Okay, just a simple way of visualizing that. All right, important unit organic molecules in organisms include glucose, triacylglycerols, phospholipids, amino acids, and nitrogenous bases. And we'll touch on all of those during this lecture. All right, we're going to start with the first of our four major biological macromolecules, the carbohydrates, the most abundant of the organic compounds in living organisms. It's not the most abundant molecule, but the most abundant organic compound. What do you think would be the most abundant molecule in every living organism? The most abundant molecule, whether we're talking about animals, plants, bacteria.
DNA? Or what?
No, it's actually water. Water, correct. Yep. Water is actually the most abundant. All right, carbohydrates have this general formula: carbon, oxygen, and twice as many hydrogens as there are carbon or oxygen. This little n here simply means a number of carbons, hydrogens, and oxygens. And you might want to write here that the smallest of the carbohydrates, actually contains 3 carbons. So that means that particular carbohydrate would have 3 carbons, 3 oxygens, but 6 hydrogens. Okay, so that's just a general formula. All right, carbohydrates function predominantly as energy storage molecules or structural molecules. 2 major functions for carbohydrates. They also play lots of other roles as well. They play an important role in cell-cell interactions and recognition, and they form part of what we call the building units of nucleic acids. We'll come back to that later on. 3 very broad categories of carbohydrates are listed here: the monosaccharides, disaccharides, and polysaccharides. Let's look at each of these in turn. So the first group are the monosaccharides. These are the simplest carbohydrates. Everyone will be familiar with these. They consist of a single molecule, so a single monomer unit. Okay, single molecules, not joined together. Okay. They're classified according to the number of carbon atoms they contain. And those monosaccharides, which consist of 5 or more carbons, are typically found in ring structures. And we'll look at this in just a second. Monosaccharides are readily soluble in water, and they have a varying degree of sweetness. All right, let's look at just 3 of the monosaccharides. Okay, let's start with this one. Everyone knows glucose. This is 1 molecule of glucose. There it is there. And now with the chemistry that you've been learning, you'll actually understand what you're seeing there. So you can see 6 carbons, 1 molecule of glucose, 6 carbons. You know what these lines represent. What are these lines in between the carbons?
Covalent bonds.
Covalent bonds, single covalent bonds. Yep. And you can see each carbon forms 4 covalent bonds. What do we have up here?
Double bond.
A double covalent bond, correct? And we can see what elements are found in a molecule of glucose. Of course, we've got carbon. What else do we have?
Oxygen.
We've got oxygen. And we have hydrogen, correct. Okay, now where you see this is just a bit of an abbreviation there. That carbon is bonded to the oxygen, and the oxygen has a covalent bond to the hydrogen. Okay, that shorthand method just places the oxygen and the hydrogen together without showing the covalent bond in between them. Okay, so that's glucose. Another example, galactose, and that's very, very similar to glucose, 6 carbons. And here we go, viticulture students are also very familiar with this one, fructose. 1, 2, 3, 4, 5, 6 carbons also. And you can see the double bond is in a different location here. Okay, so they're shown as straight chain molecules, but how they exist naturally is shown here. They exist naturally, mostly in these ring structures. Okay, and there we go. So there are our monosaccharides. Now let's look at disaccharides. So the second group. So once again, imagine our just single monosaccharides, and if 2 of them were bonded together, we would have 1 disaccharide. Okay, so monosaccharides in their cyclic form can bond together in a variety of ways, and they do this through special bonds called glycosidic linkages. And we'll talk a lot more about them in living systems too. They form a glycosidic linkage to form a disaccharide. So here's a couple of examples for you. Lactose, everyone's familiar with the main sugar found in milk. That's called lactose, and 1 lactose molecule contains 1 glucose monosaccharide bonded to 1 galactose monosaccharide. And you can see that down here. So here's 1 lactose. Sucrose, this is an important one, contains 1 glucose and 1 fructose monosaccharide. That's shown here. Okay, where do we find sucrose? Sucrose is synthesized by plants, yes, synthesized by plants, and it's actually the major type of carbohydrate that's transported within the body of the plant. And the reason for this is sucrose is not as reactive as, for example, glucose or fructose. So it can be transported through the plant without being metabolized while it's being transported. So it can get to where it needs to go within the plant. Okay, so keep that in mind about sucrose. Okay, polysaccharides, our third group of carbohydrates. Polysaccharides consist of chains of hundreds or even thousands of monosaccharides bonded together. They perform 2 major functions. One is as structural components, and number 2, they might function as storage reserves. Okay, structural or storage polysaccharides. Let's look at examples of both. Here's our first one, cellulose. Cellulose is a structural polysaccharide. So where might we find cellulose? Well, lots of cellulose in wood. Here we have some cotton fibres. And these are also composed mostly of cellulose. So remember cellulose as a structural polysaccharide. All right, let's look at the chemistry of cellulose. So as we said, cellulose consists of many monosaccharides joined together. The monosaccharide is glucose. Here we have one glucose, and that's bonded to another glucose, and so on and so on. This is the same thing here, but we're looking at a surface view. Glucose monosaccharides bonded together, and again here, and they're actually associating or bonding together laterally to form a sheet. Then these sheets, here's a sheet, this is a side view of the sheet, will actually bind or bond to another sheet. So they stack on top of each other and they form a very, very tough microfibril of cellulose, what we call a microfibril. You can think of it as a fibre if you like. And what you can see in this image here, this is an actual image of cellulose microfibrils running in different directions on the outer surface of a plant cell wall. This is what they actually look like. So that's cellulose. Remember that cellulose is composed of glucose monomers. All right, here's another structural polysaccharide called chitin. Chitin resembles cellulose in that it's made up of many monomer units joined together to form these long chains. They also associate laterally and stack on top of each other to form microfibrils. But these monomer units are not glucose. Okay, so note that they're very similar to glucose, but they're chemically different. Okay, they are not glucose monomers. All right, where do we find chitin? Chitin is predominantly found within the cell walls of fungi. So when you think about your ferment, yep, and your yeast, Saccharomyces cerevisiae, and any other yeast that you've got in there, these are tough little cells. Tough little cells because of the presence of chitin in their cell walls. Also, we find chitin in the exoskeleton of arthropods. All right, moving on now from structural polysaccharides to storage polysaccharides. And the major ones that we're talking about here, starch. So starch is a storage polysaccharide. That is, it's a form of energy storage. Starch is stored for future reserve of energy. Okay, so the major storage polysaccharide in plants is starch. Starch is a polysaccharide. What is the monosaccharide subunit? What is the monomer that makes starch? Once again, it's glucose. Yep, glucose. So note that. Just like cellulose, starch is made up of repeating glucose monomers, but the structure is quite different. All right, what about animals? Animals tend to store carbohydrates, not as starch, but as glycogen. Once again, glycogen is composed of repeating glucose subunits, just like starch and just like cellulose. Okay? Okay. Oh, it sounds like we've got some company in the background.
Yeah, I'll be sick.
All right, just a couple of nice photos here showing starch granules. All right, moving on to our next major group of biological macromolecules, the lipids. Lipids are a group of molecules which includes fats, oils, waxes, phospholipids, steroids, and various pigments. A lipid can be defined as a substance that can be extracted from biological materials using non-polar organic solvents such as ether, benzene, or hexane, but lipids can't be extracted by water. All right, don't worry too much about these terms.
I think we lost Silvana.
Yeah, I think that's. Yeah, mine's frozen up.
Yeah. Did your internet drop out again, Silvana?
Hello?
Oh,
Hi.
yep.
Welcome back.
Okay.
I'm sorry.
That's all right.
Cannot believe this is happening. Oh dear. All right, let's continue before I drop out again.
Yep.
Hopefully that's the last time.
All right. Okay.
We've lost one though.
Yeah. And I don't even think it's recording. Recording to the cloud. Okay. I think it's recording. We'll just give Liz a second to see if she can get back on.
Yep.
Oh, that's very annoying tonight.
Yeah.
Very annoying. That's the third time it's logged me out. Or it's dropped out. The internet's dropped out.
Yeah. What happened? I heard in the news something. There was a bit of a chemical spill, or something like that, in Epping. Was it?
Oh, really? A chemical spill?
Well, there was something.
There was a fire.
Oh, was there? Okay.
There was a fire next to Westfield Shopping Centre.
Oh, and there might have been. I don't know if it was a chemical spill, but I heard something about, like needing to close the doors because something might have been burning off, and Yeah.
Oh, was that at the Epping campus, you mean?
Not. Oh, I just heard in Epping. I don't
Oh,
know.
just in Epping somewhere.
Yeah,
Yeah, no,
floating
I
news,
didn't. Yeah, I didn't hear about that.
yeah. Okay.
No.
Yeah, I'm not sure. I just
No.
heard it in brief. So yeah.
Okay, yeah, there was a fire, a grass fire next to Westfield.
Yeah. Yeah.
But yeah, that's not Epping actually. That's South Morang. So
Yeah. No, because I was saying on the news that like there was about 19 suburbs, and like I'm in Bundoora, and they were saying even Bundoora like to keep the windows closed.
oh, really? When was this?
Oh, look, it was probably about lunchtime today. I heard it.
Today. Oh,
Yeah.
okay.
Yeah.
Ah, no, I didn't hear anything. But then I was teaching today, so
Yeah. Yeah.
yeah.
It mustn't have been that bad, I guess. If you were in Epping and it was bad, you would have probably.
Yeah, that's right. I would have noticed something.
That's right.
Yeah. No. Oh, maybe Liz is not coming back.
Okay.
All right, so we'll just press on.
Yep.
Okay, so lipids. This table gives a nice summary of the different functions of lipids. They're really, really diverse, and they're involved in so many different processes in biological systems. So you can see here they function as in storage. Oh, here we go. This is back. Storage. They have protective functions, structural, informational, pigments, photoreceptors, and cofactors in biological reactions. So this is just for your interest here. But because they're so diverse, we can't actually describe lipids in terms of a single type of monomer subunit. Yeah, they're very varied in terms of both function and structure. All right, so let's start by looking at some of the simple lipids.
Uh-oh.
Again.
No, that was quick. We didn't even get 2
Terrible.
pages.
How ironic on the one that she wanted to cover most content on.
Yeah, well, yeah, that's right. It's pretty full on, this one.
Yeah.
Although there's not a lot of there's probably more memory material than actually needing to learn formulas or anything like that.
Yeah, that's true.
Did you leave the meeting before, Darren, or?
No, I stayed on.
Oh, okay. Yeah. So when she gets back on, yep. I'd actually left. I thought might have to reconnect.
The first time when she wasn't a host, I think
Yeah.
the room left entirely, so she must have closed it, and that's when I found out. But this last one, I just stayed.
No. All right. Well, I won't. It's easier to just stay on, I think.
Absolutely.
All right, I think I'm going to have a lot of problems tonight. Okay, who wants to persist with this? Or
Yeah.
shall we just...
I don't mind, but yeah.
Yeah. Look, what I might suggest, if this keeps dropping out on me, which it looks like it's going to, I'm not going to come back in again. What I'll do is post a recording of this lecture.
Yeah.
Okay? Yeah, because otherwise this could keep going on and on.
All night.
All night, yeah.
All
All right, so if this drops out on me one more time,
Yep.
yeah, don't bother coming back in, and I'll put a post on Moodle as soon as I get a lecture recording of this up, okay?
No
Yep.
worries.
Okay.
All right, you happy with that, Darren and Liz?
Yeah, no issue.
Yep. No worries.
Okay, great.
Pretty frustrating.
Yeah, so sorry, very frustrating.
No, that's right.
Yeah, I need to find out why this is happening. All right, so what we were saying, fatty acids. Yep, fatty acid chains that contain only single covalent bonds are called saturated fatty acids, and those that contain at least one double covalent bond, as you can see here, are referred to as unsaturated fatty acids. Let's look at some examples of these. All right, here we have an example of a saturated fatty acid. So you can see it has a very long chain of carbons, 18 carbons, and this particular example of fatty acid is called stearic acid, and you would find this predominantly in animal fats, not exclusively, but predominantly in animal fats. There are only single covalent bonds between the carbons in this long chain. This is called the carbon backbone. Don't worry about this part up here. So that's a saturated fatty acid. Here's another example of a fatty acid. This is called oleic acid. It's got the same number of carbons, 18 carbons in the carbon backbone, but the main difference here is the presence of one double covalent bond, and that makes a big difference. And we'll talk about that a lot more in living systems too also. All right, so this is a, yes, sorry, go on.
The word saturated versus unsaturated, what does that relate to?
Yes, well, that actually relates to when we say that, that the fatty acid is saturated, that's actually relating to these carbons being saturated or surrounded by the maximum possible number of hydrogens.
Okay.
That's what we refer to as saturated. Unsaturated, so if you think about these carbons here, yep, on either side of the double covalent bond, those carbons don't need to attach to as many hydrogen atoms.
Got it.
So they're not surrounded by the maximum possible number of hydrogens.
Thank you.
Okay? And hence the term unsaturated. Okay, so they're fatty acids, and you'll find that many of the, not all of them, but many of the other groups of lipids actually contain one or more fatty acids. Right, oh, get rid of that. Sorry, go on. All right, let's move on to waxes. So we're still talking about simple lipids here. Waxes are totally insoluble in water, and that's because they tend to have very large non-polar components. Where do we find waxes? They occur as coatings on the surfaces of plants. So we have this waxy covering called the cuticle, and that prevents water loss or limits water loss from the plant by evaporation. But also waxes also feature in animals as well, as you can read there. All right, our next group of simple lipids are called triacylglycerols, also referred to as triglycerides or simply TAGs, T-A-G. Triacylglycerols, also referred to as neutral fats or oils. These are the fats that tend to be stored in special storage cells of adipose or fatty tissue in mammals, and also in cells certain plants. So, for example, in seeds, castor bean, canola, coconut, oil palm, flax, and also some fruits, so avocado and olives. All right, and here's a very nice image of some fat cells found in animals, and these cells are called adipocytes. Moving on now to complex lipids. Complex lipids are critical as they form components of biological membranes. When we say biological membranes, we're actually referring to membranes that enclose cells of every living organism, whether they're plant cells or animal cells, or if we're talking about a yeast cell. All cells of living organisms are enclosed in these special membranes. And lipids, complex lipids, feature very heavily in these membranes. All right, and here they are, the all-important phospholipids. Let's look at the structure of a phospholipid. Okay, here we have one phospholipid molecule, and we can divide a phospholipid into two regions. Here's the phospholipid head, and you can probably appreciate what you see in there because of the chemistry that you've learned. So you might recognize this one here. This is a phosphate group, and you might recall that from that table of polyatomic ions from lecture 3. So there's a phosphate group which is bonded to this group of atoms here, and you can see a combination of both single and double covalent bonds. This group is actually referred to as glycerol. Everyone's familiar, or at least have heard of that term, glycerol. All right, so what's special about this head region of the phospholipid? It's actually hydrophilic. What do we mean by hydrophilic? We mean water-loving. In other words, this part of the phospholipid likes to associate or interact with water molecules. Okay, hydrophilic, remember, water-loving. Then the next part of the phospholipid molecule is shown here by what we call the phospholipid tails. And these two tails are actually fatty acids. Two fatty acid molecules attached to the actual head. And fatty acids, of course, being structured the way they are hydrophobic. What does that mean? Hydrophobic?
They don't like water.
They don't like water. Yep. So hydrophobic means water fearing or water hating. So the fatty acid tails of the phospholipid molecule will try to escape any interaction with water molecules. So this type of molecule that has both a hydrophilic portion and a hydrophobic portion is termed amphipathic. So a phospholipid is an amphipathic molecule. Okay, we're still going. Now, I'll just skip this slide just for a second to show you what happens when we place phospholipids. There's one phospholipid, and let's imagine placing them in water. So here's the water here. What are the phospholipids doing? You can see that they're all upside down. Of course, because the head regions like to interact with the water, they're hydrophilic, and the tail regions shown up here are trying to escape any interaction with water molecules because they're the hydrophobic component. Now, what do you think would happen if we force those phospholipids into the water, so agitate the actual solution? The phospholipids will form this spherical structure, which is referred to as a mycele. So once again, the hydrophilic heads of the phospholipid interact with the water molecules, but the hydrophobic tails will try to escape by aligning themselves towards the center of that sphere. All right, and finally, and this is the important one, this is what phospholipids do. This is how they arrange themselves to form a biological membrane. So remember those membranes that enclose all living cells. Look at what we have here. 2 layers of phospholipids. 2 layers, we call this a bilayer, a lipid bilayer. And once again, look at how those phospholipids are arranged. This outer layer of the lipid bilayer, the heads are interacting with water outside the cell, for example. And this inner leaflet or inner layer of the lipid bilayer, the heads, the phospholipid heads, interact with the watery interior of the cell. And again, the tails are all positioned towards the inside of that lipid bilayer to escape any interaction with water. All right, now we can go back one slide and just review what we've looked at. So phospholipids in water can rearrange themselves to form a self-sealing closed vesicle whose surface is a bilayer. The hydrophilic heads of the outer layer of the bilayer are in contact with the external aqueous phase, so the water outside the cell, or solution outside the cell, and the hydrophilic heads of the internal layer of the lipid bilayer contact the water to the inside of the cell, so the internal aqueous phase. Then the interior of that lipid bilayer is formed by association of those hydrophobic tails of the phospholipids. Phospholipid bilayers are the basic structural units of all biological membranes. So you can appreciate the importance of those. All right, our next group, the proteins. The building blocks of proteins are called amino acids. So remember back to our monomers and polymers. So here we can talk about individual different monomers. These are different amino acids, and when they bond to each other, we have a polymer, and that polymer is referred to as a polypeptide chain. So it's not quite a protein yet. There's a bit more involved before the polypeptide chain can become a fully functional protein. All right, major amino acids. The component amino acids, there are 20 of them, and we'll look at those in just a minute. 20 different amino acids. They all have the same core structure, which is shown here. The same basic structure to all of those 20 amino acids. Let's look at that. Our central carbon with its 4 covalent bonds. We call this the alpha carbon. That carbon is bonded to a single hydrogen here. Bonded to the left here to a nitrogen, which in turn is bonded to 2 hydrogens. Collectively, we call this group here an amino group. Not sure why they call it an amine group. We call this an amino group. And then to the right of the alpha carbon, we have another carbon, oxygen, double bond, and single bond to an oxygen, which is also bonded to a hydrogen. Okay, this group together is referred to as the carboxyl group. All right, so what we've just looked at here is found in every amino acid. Every amino acid has the alpha carbon, hydrogen, amino group, and carboxyl group. Where those 20 amino acids differ from each other is here, in what bonded here to the alpha carbon. This is a variable group called the R group, or we call it a side chain group. And I'll show you the different side chain groups in just a moment. All right, this table shows how proteins are classified. You don't need to remember this. This is for your own interest. Okay, proteins can be classified according to biological function. You can see a whole host of different biological functions. They can be classified according to composition or according to solubility. All right, here we have the first 10 of the 20 standard amino acids. So let's look at this in just a little bit of detail. So remember I said that every amino acid differs in terms of its R group, that variable group or side chain group. Here's an example. So the R group or side chain group in this amino acid, which is called glycine, is simply a hydrogen. This is the smallest of the amino acids. Our next amino acid is called alanine, and its R group is CH3. Valine has this as its R group, and so on. So you can see that all the R groups are different from each other, all highlighted here in yellow. Remember also that the core structure of all the amino acids is exactly the same. All right, what elements can we see that make up amino acids? What elements?
Carbon, oxygen.
Carbon, yes. Carbon, oxygen.
Nitrogen.
Nitrogen.
And hydrogen.
Hydrogen, yep, so they all have these elements, and a couple of them have a different element.
Sulfur.
Sulfur, yep. So methionine and cysteine also contain sulfur. There we go. Just clear that. And here we have the rest of the standard amino acids. And of course, those side groups are highlighted there in different colours. All right, now we're going to look at the different levels of structure within proteins. We can refer to the primary structure, secondary, tertiary, and quaternary structure. Let's start with the primary structure. The actual sequence or order of the amino acids in a polypeptide chain actually defines what's called its primary structure. And this different order of amino acids in different polypeptide chains will actually give different biological properties to specific proteins. Okay, that's all you need to know. Primary structure refers to the order or sequence of the amino acids within the chain. Okay, secondary structure. What's meant by secondary structure? Secondary structure is actually determined by the arrangement of those variable R groups or side chain groups on either side of that polypeptide chain. And depending on how they're arranged, one or both of these different patterns can be seen. One's called the alpha helix pattern or motif, and the second is called the beta sheet or beta pleated sheet. Let's look at those here. So what we have here is called the beta pleated sheet. So here we have one chain of amino acids, and here we have another chain of amino acids. And the way they interact with each other, and what you're seeing here are hydrogen bonds between those two chains, actually pulls, oh, someone's gone, actually pulls the polypeptide into this zigzag arrangement, which is called a beta pleated sheet. And these blue arrows that you can see here, they're just a shorthand schematic of beta pleated sheets. All right, the second most important secondary structural pattern or motif that you'll see is here. This is called the alpha helix. And the schematic that shows that is simply this coil here. Okay, so that's secondary structure. Up here we see what's referred to as primary structure. It's simply a chain of amino acids. The sequence of, so which amino acids are found in this polypeptide chain. All right, next we have the tertiary structure, the level of tertiary structure. And tertiary structure refers to the final folded three-dimensional shape of the polypeptide. And this is due to side chain interactions, but keep in mind that these interactions may be between side chains that are found very, very far apart from each other in the actual polypeptide chain. Let me just illustrate this. So you may have a very long polypeptide chain. Okay, so I'm not going to draw all of them. And it may be an interaction between this one here and this one very far away from it in that sequence of amino acids that actually determines this folded three-dimensional shape. All right, so just keep that in mind. All right, and what we can see here, this is an example of a folded polypeptide, and it consists primarily of the alpha helix structural motif. Here's another example of a folded polypeptide, and you can see by the presence of all those blue arrows that it contains mostly beta-pleated sheets. Third example here is a combination of both alpha helices and beta sheets. All right, finally we have our quaternary structure, fourth level of organisation. And the quaternary structure refers to the association of two or more polypeptides together to form the final functional protein. This is how many proteins become functional, only when two or more polypeptides actually associate with each other. So this particular example here shows 1, 2, 3, and 4 polypeptides. 4 separate polypeptides associate together to contribute to the final quaternary structure of a protein. All right, some examples of proteins that are shown here. We've got keratin, silk, collagen, and elastin. All right, here we have a variety of keratin forms. So in the fur of this possum, feathers, in the shell, and spider web. All right, our final group of biological macromolecules are the nucleic acids. And remember we said at the beginning of this lecture, the two major types of nucleic acids are DNA and RNA. Nucleic acids are found in all living cells. This is part of what defines a cell as being a living structure. DNA and RNA. All right, we won't talk about the function of DNA now. We'll save that for a later lecture. But I know that you're all familiar with the concept of DNA. DNA are the molecules in which information on cell structure and function is stored and transmitted from one generation to the next generation. So the nucleic acid called DNA, this is the molecule that contains what we refer to as genes. And those genes are passed on to offspring, regardless of whether we're talking about animals or plants. Okay, it applies equally. Now, let's consider this fact. All the cells, all your cells, or all the cells of a plant contain exactly the same DNA. Exactly the same. So why is it that some cells are structured differently from other cells, or they function differently from other cells if they all have the same, exactly the same DNA?
It's the protein groups that are attached to the DNA that give it different functions, is that?
Sort of, yeah. But it's actually the genes encoded within that DNA that are expressed differently in different cells. So just to put it really, really simply, some of those genes found in the DNA will be switched on in some cells and not other cell types. Yeah?
Yep.
Or there are certain genes that are switched on or expressed, we say expressed at really high levels, and others that are expressed at really low levels. Some are expressed only at certain stages of development and not others. And then you've got this really complex mechanism of regulation of gene expression. Okay, so that's the simplest way of looking at it. Differences in what genes are expressed and when they're expressed. Okay, but the DNA itself is exactly the same. Okay, and we'll talk more about that later on. Okay, the building unit, the monomer unit of both DNA and RNA is the nucleotide. So once again, the monomer is the nucleotide, and if those monomers bond together, then we have the formation of a polymer. In other words, a chain of nucleotides to form a strand of DNA, a chain of DNA, or we can say a chain or strand of RNA. All right, the nucleotide has three components: a nitrogenous base, a monosaccharide, which might be either ribose or deoxyribose. I'll come back to them. And a phosphate group. There's that phosphate group, that polyatomic ion once again. All right, let's look at the parts of a nucleotide, which we can see here. Once again, three parts: a nitrogenous base, a sugar or a monosaccharide, and the phosphate group. Now we can look at the actual chemical structure of a nucleotide. Here we have a nitrogenous base, and you can see why it's referred to as being nitrogenous, because it contains lots of nitrogen. Okay, attached to that base is a monosaccharide. This monosaccharide is either ribose, if this nucleotide is found in RNA, or it's deoxyribose, which is almost identical to what we're seeing there, if the nucleotide is a part of DNA. Okay, so that's a monosaccharide here. So pictured here is ribose. It will be slightly different, as I said. That is, it will be deoxyribose or 2-deoxyribose if it's part of a nucleotide found in DNA. Okay, this sugar component is bonded to a phosphate group, okay, which you're becoming more and more familiar with. So that's a nucleotide. All right, nitrogenous bases are of two types. There are pyrimidines and there are purines. Not particularly useful to remember those names, but all it means is that the pyrimidine bases are comprised of a single ring of carbon and nitrogen. Let me just go back. So we're talking about the nitrogenous base. If there was only one ring instead of two rings, then that would be classified as a pyrimidine. So there are three of these: uracil, which is abbreviated to U, cytosine, C, thymine, T. Okay, so three pyrimidines. And then we've got the purines. These are nitrogenous bases that contain two rings, adenine A and guanine G. And I'll just go back. And of course, this one is adenine, this example here. All right, let's look at the differences between DNA and RNA, our two major types of nucleic acids. So as we said earlier, in DNA, that sugar group, the monosaccharide, is 2-deoxyribose. The bases, the nitrogenous bases found in nucleotides of DNA are adenine, thymine, guanine, and cytosine. RNA has a ribose monosaccharide instead of a 2-deoxyribose. And the nitrogenous bases found in nucleotides of RNA are adenine, uracil instead of thymine, guanine, and cytosine. Okay, so you can see the difference there. DNA has these 4 nucleotides, combination of those 4. RNA polymers have a combination of these 4 nucleotides. All right, DNA exists as 2 long chains of nucleotides running in opposite directions, and they're wound together into a double helix formation. RNA, on the other hand, exists as 1 chain, 1 single chain. Okay, another difference between DNA and RNA. All right, so here's our last slide, and we'll finish with a closer look at the structure of DNA. All right, so this representation here is showing us that double helix formation of DNA. This is the same thing here in B, but now we can more clearly see what's referred to as the backbone of a chain of DNA, a strand of DNA, and here's another backbone. In other words, another chain of DNA. We can also see here how the nitrogenous base components are actually found towards the inside of that DNA double helix. All right, let's enlarge just this region here so we can look at it in a little bit more detail. Okay, here it is enlarged so we can see the actual chemical structure. And we're going to start by looking just at one chain or one strand of DNA, which is found here. And we can see that there are 2 nucleotides. Here, there's one nucleotide there. This one has a cytosine base, so we just refer to this nucleotide as cytosine. The second nucleotide here is thymine because it has a thymine base. All right, so you can see that the cytosine is attached within the chain to this thymine. Here's the attachment here. And that bond or that link between nucleotides in the chain, in the same chain, is called a phosphodiester linkage. So nucleotides are bonded to each other via phosphodiester linkages. Let's look at the opposite strand or opposite chain of DNA here on the right, and this one also consists of 2 nucleotides, but they should be connected to each other. It's not showing a connection here. All right, they're also connected to each other via phosphodiester linkages. And you can see that these are upside down, these nucleotides are upside down relative to the nucleotides in the other chain. This is what we mean when we say that they're opposite. The 2 chains or 2 strands of DNA are opposite to each other. Okay, nearly there. All right, so how are the 2 chains or 2 strands actually attached to each other? How are they bonded together?
The 2 the amino acids, is that hydrogen bonding of the?
Yes, absolutely correct. So you can see this dotted line here, and this shows a hydrogen bond here, another one there, and another one here. So we've got 3 hydrogen bonds forming between the cytosine nitrogenous base and the guanine nitrogenous base from this nucleotide here. Yeah, 3 hydrogen bonds there, and here we have a hydrogen bond there, and we've got one here. So you always see 2 hydrogen bonds between a thymine and an adenine, and 3 hydrogen bonds always between cytosine and guanine. And this also explains why cytosine always forms a bond to guanine, and thymine always seems to pair up with adenine on the opposite chain. Okay, so remember that hydrogen bonds actually keep the chains connected to each other, whereas the phosphodiester linkages are the bonds that join nucleotides together within a single chain. And of course, remember, we're only looking at a couple of nucleotides on each chain here, but in fact, each chain of DNA or each strand of DNA contains millions and millions of nucleotides. Okay, and that completes our lecture.
All right, we got it. We got there.
We did. We actually got there in the end, with a lot of interruptions.
Oh,
Yeah.
that's all right.
Okay, but we did get there. Very late, but we got there.
So the lectures from now on, we're going to always cover two.
Most weeks,
Yeah,
yes.
okay, all right.
Most weeks we'll be covering 2
Yep.
lectures.
So we're ramping up now.
We're ramping up.
Yep. All right. Yep.
We started slow for good reason.
Yeah.
But now everyone's comfortable, familiar with the system.
Yep.
You've got the fundamentals under your belt, so we can really start ramping up the subject material.
Yep. Yep.
All right.
Okay.
And
All right.
before we go, tutorial questions for this week. Okay, chemical reactions and biological molecules, tutorial number 4. And these are assessed.
Yep.
Okay, and due, of course, next Monday by 6 p.m.
Okay. Yep.
Okay, if you've got any problems with the tutorial questions, anything you're not understanding, please let me know.
Yep. Okay. All right.
All right.
All right.
Okay.
Thank you.
Thank
good night.
you. Have
Bye.
a good night. And
Susanna.
yep, see you next week.
Okay. No worries. Bye.
All right. Thanks. Bye-bye.